Hi everyone, my name is Arushi Madan and I'm from Tesoro High School.
Hi everyone, my name is Shreya Madan and I also go to Tesoro High School.
My name is Araya Benavides and I go to Downtown Magnets High School. And today we'll be covering solutions that can be designed by identifying misfolded proteins in patients with Parkinson's disease.
Parkinson's disease gradually disrupts the person's brain, which causes shaking, stiffness, difficulty with walking, balance, and coordination. Patients with Parkinson's also lack norepinephrine, which is a main chemical messenger of the synaptic nervous system. The protein structures of someone with Parkinson's are misfolded into amyloid fibrils proteins. Misfolding and inclusion body formations are common events in neurodegenerative diseases characterized by deposition of misfolded proteins inside or outside of the neurons.
Our question relates to identifying the issue before it occurs. In this case, our goal with our proposal is to identify when the proteins in the human body start to misfold in order to design possible solutions such as an antibody to prevent this disorder. Techniques such as cryo-electron microscopy can provide 3D images of these protein structures. Previous research has shown that misfolded proteins form clumps of proteins known as Lewy bodies. A team of researchers at the University of Pennsylvania injected the misfolded alpha-synuclein into a part of the mouse brain rich in dopamine-producing cells. Lewy bodies began to form. Thus, Parkinson's disease began to develop in the mice. However, not much research has been done on the idea of identifying the misfolded protein beforehand and attempting to prevent it from misfolding. Currently, doctors are utilizing single photon emission computerized tomography scans to identify if a patient has Parkinson's disease.
In terms of social context, Parkinson's disease affects 60,000 Americans each year and 10 million people globally. Generally, people start to develop Parkinson's at the age of 50 and above. It is rare to see development before this age. Many patients are suffering with this disease in their daily lives and cannot live normally even by themselves. In 2013, the Obama administration launched the Brain Research through Advancing Initiative Neurotechnologies Initiative, or also known as the Brain Initiative. This initiative is aimed to achieve the development and application of innovative technologies that can create a dynamic understanding of brain function. It began to one day cure brain disorders such as Parkinson's and Alzheimer's disease. Furthermore, researchers have established how a protein called alpha-synuclein, which is closely associated with Parkinson's disease, functions in healthy human brains. By showing how the protein works in healthy patients, the study offers important clues about what may be happening when people develop the disease itself. Parkinson's disease is one of a group of conditions known as protein misfolding diseases, because they are characterized by specific proteins becoming distorted and malfunctioning. These proteins then cluster into thread-like chains which are toxic to other cells. The researchers tested mutated forms of alpha-synuclein that are linked to Parkinson's disease. This was found to interfere with the same mechanism, essentially by impairing the ability of alpha-synuclein to regulate the flow of synaptic vesicles and hence compromising the signaling between neurons.
According to scientist Sarah S. and her team at the University of Basel in Switzerland, they're using light and electron microscopy on the brain tissue from 5 Parkinson's disease brain donors to identify the alpha-synuclein or protein structures. Also, scientists are able to use STED microscopes to confirm that the Lewy bodies consist of crowded structures and dysmorphic structures. This technique also allowed scientists to discover a protein-lipid compartmentalization. Scientists from Harvard University are currently taking skin cells and converting them into stem cells. Stem cells can be converted into any tissue in the body, including brain cells. Hence, with this technique, scientists can observe the Parkinson's and other neurodegenerative diseases because stem cell-derived neurons can help them study this disease. But even with that current research being done related to Parkinson's disease, we still do not have a cure.
We believe that using both the atomic force microscopy and cryo-electron microscopy simultaneously to take the image of the protein before degeneration and compare it with images of healthy protein structures in order to understand the differences between the two structures will be beneficial for curing this disease. Atomic force microscopy, also known as AFM, uses a probe that scans the surface of the object being studied and allows scientists to obtain the mechanical properties of the sample. Additionally, the cryo-electron microscope is an alternative approach to X-ray crystallography. This means this technique can scan macromolecular structures without the need of crystallization. Once the image is captured, then a high-resolution 3D model can be designed of the protein, similarly to what is shown on the screen. We believe that if these two methods are combined, possibly a cryo-EM AFM, then scientists could develop a highly advanced image of the proteins. The image would allow scientists to create a drug or an antibody that can be injected into the body before the proteins are misfolded and damaged to prevent these complications from occurring. Also, since the AFM has a tip, an antibody could be attached to the end of the tip and unfold the misfolded proteins, then possibly realign the proteins back together.
The challenge to the proposal is that once we freeze the protein sample during cryo-electron microscopy, the probe of the atomic force microscope will not be able to scan the frozen sample.
In order to resolve the problem, we propose to add heaters in the area where the protein is being scanned in order to melt the water. Then add antibodies to a substrate to make sure that the sample does not flow off with the water, as well as add antibodies to the probe in order to prevent the proteins from misaligning.
In conclusion, in order to try a new perspective for the study of Parkinson's disease, our proposal suggests that the combination of the atomic force microscope and the cryo-electron microscope will be beneficial to capture a high-resolution image of the protein right before it starts to misfold. This will allow scientists to further enhance their understanding of the misfolded proteins that are responsible for Parkinson's disease and possibly create a drug or an antibody in the future to prevent the proteins from misfolding. Once a potential cure is created, scientists can test the drug on rats or any other form of animal that has a similar brain structure to humans and eventually introduce it to humans. This will hopefully cure Parkinson's disease. Thank you.
All right, questions?
So you said the proteins are misfolded. Would applying the antibodies unfold the proteins? Is that the path?
Yeah, so the atomic structure or atomic force microscope and the cryo-EM are hopefully our proposal takes a clearer picture of the protein. So then once we inject the antibody, whether it's to the protein or when we discover the structure, we can propose an antibody that will refold them back to normal.
Okay.
Students, do you guys have any questions?
So you're interested in sort of understanding the onset of this misfolding, right? What's the where do you plan to source the proteins from, right? Like, how do you plan to get proteins that are properly folded and misfolded for this set of experiments that you have, right? Because you need samples, right? Are these going to be purified samples? Are you going to get them from patients? And then once you have these proteins, how do you get them over this time course, right? Is it just a matter of many experiments and hoping that you find one that's starting to misfold, or what's the approach there?
So we were, as we were researching, we found out that the decrease of dopamine levels actually cause Parkinson's disease. So whether we inject the lower levels of dopamine levels in rats, let's say, then we could their protein structures will automatically start to misfold because that's the leading cause of Parkinson's disease. So we could obtain the samples from rats or whatever organism we test the low levels of dopamine.
And then the time period.
Oh, and can you repeat the second part of your question? Was that all of it? Oh, okay.
Okay, so you're talking about trying to combine the utility of the AFM and the electron microscope,
Yeah.
right? So the way you proposed it, there's a bit of a problem. First, what do you think is potential problem with trying to heat up a sample that's frozen in an electron microscope?
Well, we ran into that problem. Like, if we freeze the protein structures, then we can't actually obtain the image of the protein itself because it'll be frozen and the tip will not be able to go through the frozen sample.
Right.
So if we heated the sample, then the water could flow off and we'd have actually a substrate with an antibody attached to it. So that way the actual sample of the protein can stick onto the substrate and not flow off with the water once it's heated and melted
I
off.
think that's a very interesting question, and whether the protein could stay there, that's a very interesting thing, that the protein could even be suspended in the vacuum, but where's that water going to go inside of that electron microscope? It's in a vacuum, it's going to evaporate, and those engineers are going to be very unhappy. On the other hand, if you do the AFM first off the fresh sample, then you could do it in your device if you built a device to freeze it in place.
Okay.
Right? So
Yeah.
if you reverse the direction of those two, you actually could make it perfectly compatible, right? And
Okay.
the second thing I would ask is, if you're talking about putting an antibody on a tip of an AFM and then trying to capture a protein like the Lewy bodies, those type of proteins, what happens if you put that into a cell that doesn't have Parkinson's? Is that protein still there? And are you just going to get a bunch of loose protein on the tip? Are you pulling on like an individual protein, or are you planning on pulling on the body itself? Could you clarify that just a little bit?
We're planning to pull in the body.
Yeah. Yeah, because if we were to do it in like a healthy cell without Parkinson's, the proteins wouldn't be folded. So we are planning to do it on the body itself.
Thank you.
Thank you.